Welcome back, guys. So today we are discussing this important topic in the cellular physiology, that is membrane transport. In this membrane transport, first we are going to see the classification. This classification is membrane transport is based on energy usage. Based on this energy usage, this classification of membrane transport. So we're going to see the mnemonic. This energy, this currency of energy in our body is ATP. This ATP is the energy currency for body. So this A stands for active transport. So it stands for active transport. Here it requires ATP. Active transport requires ATP. With simple understanding, we all know these terms. So P stands for active transport. P stands for passive transport. E stands for passive transport. Here no need of ATP. This active transport requires ATP, and passive transport there is no need of ATP. So I want to discuss one by one. First, here is active transport. Active transport. So, what is this active transport going to see? Suppose this is a membrane. This is the membrane thickness. This is the outer of the membrane. This is the inside of the membrane. The outer and inner. So these are these molecules. This molecule's concentration is high concentration in the outer side of the membrane. The inner there is very low concentration of molecules inside this membrane. So across this membrane, the molecules move from high concentration to low concentration, high concentration to low concentration, high to low concentration. So this is the movement of molecules. This movement of molecules from high to low is the simplest form of all. It is called simple diffusion. Excuse me, we are discussing here is the passive transport. Sorry, I got confused a bit. We are discussing this passive transport. Here the first one is simplest of all, simple diffusion. Simple diffusion is the simplest form of all. Simple diffusion. Example: diffusion of gases across the respiratory membrane. The respiratory membrane. What are the respiratory gases? It's simple: oxygen and carbon dioxide. So oxygen and carbon dioxide passes through the diffusion. This diffusion process across the respiratory membrane, in and out of the across the respiratory membrane. In the simple diffusion, as you see, no ATP is required here. No ATP is required here. Substances move from high to low concentration. We have established this fact here. Substances move from high to low concentration. Now this diffusion depends on Diffusion depends on several factors. First one being concentration gradient. Simple to understand, we have seen our concentration gradient, that is from high to low concentration. So diffusion is proportional to gradient. This we have established above. Second factor diffusion depends is diffusion is proportional to area of membrane available. This diffusion is proportional to area of the membrane available. For example, this is the membrane, so this is the area of membrane. The third factor on which diffusion depends is diffusion is inversely proportional to thickness. So this is the membrane we have already drawn. Now for example, this membrane becomes this, so this is the thickness of the membrane. These two things are self-explanatory. Increase in area increases the diffusion, area of membrane available increases the diffusion, and thickness, it actually obstructs the process of diffusion. Diffusion is inversely proportional to thickness. Self-explanatory things, nothing much to understand here. So from these three factors, concentration gradient, area and thickness, we now derive a conclusion called Fick's law of diffusion. Fick's law of diffusion. What is this Fick's law of diffusion? It states that diffusion is proportional to the gradient, that is Ci minus Co, concentration inside minus concentration of outside, multiplied by area of the membrane divided by thickness of the membrane. Diffusion is proportional to gradient, proportional to the membrane, inverse proportional to thickness. Diffusion is proportional to membrane, proportional to area, inverse proportional to thickness. So we've seen here the substance moves from high concentration to low concentration. This is Ci inside, this is Co. But here in the formula we have applied this Ci minus Co. Ci is the low concentration, Co is the higher concentration. So we introduce minus sign here. So this is the Fick's law of diffusion, proportional to minus sign Ci minus Co, A by T. Also, this diffusion is proportional to solubility. Solubility in which in lipids. This diffusion is inversely proportional to size of the molecules. This is a well established fact. The diffusion is proportional to solubility of the lipids. More the substance is soluble in the lipids present in the cell membrane to be exact, the more it is diffusible, and the bigger the size of the molecule, the less is the diffusion. As the bigger is the size, there is more hindrance to the passage of the molecules. So these three are important from the perspective of diffusion across the cell membrane. Now we are going to see some clinical scenarios. In the situation of emphysema, so emphysema is a very common occurrence. The lungs are affected in the individuals. In this emphysema, there is destruction of respiratory membrane. There is destruction of respiratory membrane, which leads to decrease in membrane Remember membrane surface area. Due to destruction of respiratory membrane, there is decrease in membrane surface area, which leads to decreased diffusion of gases. It's simple stuff. Due to decrease in membrane surface area, due to destruction of respiratory membrane, there is decreased diffusion of gases. In this condition of emphysema, there occurs respiratory problems. Another condition in the lungs, that is pulmonary fibrosis. First you see emphysema, then this condition of pulmonary fibrosis, there is increased thickness of membrane. Here actually the thickness of membrane increases multifold, which leads to decreased diffusion of gases. We have seen the diffusion of gases is inversely proportional to thickness. Inversely proportional to thickness. So increased thickness of membrane leads to decreased diffusion of gases. Next condition, pulmonary fibrosis. So I have seen from these two examples that the outcome is the same. It is decreased diffusion of gases across the cell membrane, but the causes are different. The cause in the emphysema is decrease in membrane surface area. In the pulmonary fibrosis, there is increased thickness of membrane. Emphysema, decrease in membrane surface area. In pulmonary fibrosis, increased thickness of membrane. You can also understand from this word, this pulmonary fibrosis, there is increased growth of the respiratory membrane. There is excess growth of the tissue of the lungs, which leads to increased thickness of the membrane and decreased diffusion of gases. Emphysema, decrease in membrane surface area, decreased diffusion of gases. So completed simple diffusion. Next I want to discuss is before going to the name, we are going to see through a simple diagram. So this is a membrane. This is the outer. This is the inner part. So the basic concept, there is not much to understand here. You already know from class 12. This molecule's concentration is high outside, then concentration molecule is low inside. CO, CI, concentration outside and inside. Here, passage of the molecules, simple diffusion is not possible. Not possible. Across the membrane, passage of the molecules is not possible. What we require here is a transport protein. We require here is transport protein or a carrier protein. It's carrier protein to facilitate the diffusion. This diffusion process cannot take place on its own, not through the membrane. Across the membrane, it requires a carrier protein or a channel protein to facilitate the diffusion process. This process is called, very simple, facilitated diffusion. This process is called facilitated diffusion. In name, which facilitates the diffusion, facilitated diffusion. I'm going to see some examples here. Transport of glucose, the glucose transport, it occurs through this transport proteins, GLUT. These are simple glucose transporters. GLUT stands for glucose transporters. They help in the transport of glucose across the membrane. There is also this water transport of water across the membrane through these proteins, aquaporins. These are small pores in the cell membrane, transport water. So aquaporins, the water transport, called facilitated diffusion. No ATP is required here. So process of diffusion, but it is mediated through a carrier protein which facilitates it, but this process does not require ATP. I get confused here sometimes, so be careful here. Third, we are going to see a special type of diffusion. Third, we are going to see a special type of diffusion. So again we are going to draw a membrane. This membrane is the outer, is the inner, as always. For example, there is a molecule of ammonia. So ammonia is in non-ionized form. So ammonia outside is in non-ionized form. This non-ionized form can easily diffuse. Non-ionized form of ammonia can easily diffuse. After going inside, it combines with a proton H plus ion to form ammonium ion. That is ionized form. Non-ionized form ammonium ion, ionized form after combining with a H plus ion. So this form of transport is called non-ionic diffusion. It's another type of diffusion, non-ionic diffusion. They're seen in renal system. Renal system, where transport of ammonia is a very important process. It's non-ionic diffusion. It's non-ionized form is transported across the membrane, finally converted to ionized form. Is seen in renal system. For example, one example we've seen is ammonia. Example is absorption of salicylates. Also movement of ammonia ions. Seen above, movement of ammonia ions and absorption of salicylates are example of non-ionic diffusion. You need to remember here. These two examples important. So, to see fourth type of acid movement. We have sodium ion, which is the solute. We have water, which is all obviously solvent. Sodium is the solute and water is the solvent. Now in this cell, sodium ion enters. This sodium ion is osmotically active. To understand this word, osmotically active. Why sodium ion is called osmotically active? Because along with this sodium, molecule of water also enters. Whenever this sodium enters a cell, the molecule of water also enters the cell with it. So whenever sodium enters or wherever sodium enters, water follows it. Water follows it. Wherever sodium enters, water follows it. So this water is called Hutch dog for sodium. An important concept is water is called Hutch dog for sodium. Why? Wherever sodium goes, water follows. So this sodium is osmotically active substance. So going to see this is a membrane. This membrane here, concentration of sodium is high and water is low, and here concentration of sodium is low, water is high. In this membrane, a special type of membrane called semi-permeable membrane. It's a semi-permeable membrane, that is, it permissive to passage of certain substances and not so permissive to passage of other substances. So through this semi-permeable membrane, this water here is in high concentration. This water, as you've seen, this is the solvent. Very important concept you are going to see. This is the solvent. Solvent, that is the water, moves from high concentration to low concentration. Movement from high to low through this semi-permeable membrane. This solvent, that is water, movement from high to low concentration through the semi-permeable membrane. What we call this? This is called as the universal, yes, osmosis. So osmosis is movement of the solvent. We need to remember, again I am repeating, solvent, not solute. Solvent through from high concentration to low concentration through a semi-permeable membrane. These three are important. These two are the important things we need to remember: solvent and semi-permeable membrane. So this water moves through this semi-permeable membrane from high to low concentration. Now we apply a force. Force is applied here. Force is applied. What is this force applied? The force is applied to stop the movement of water, that is, stop osmosis. Stop the movement of water, that is osmosis. This force is called as osmotic pressure. An important concept, remember. So osmotic pressure is the force which is applied against the movement of water or any solvent. In that case, it is osmosis. Remember, only the solvent moves, not the solute, through the semi-permeable membrane. The force we apply to stop this movement of solvent is called as the osmotic pressure. So we have seen this sodium is osmotically active. Why? This drags water. This water is known as the H-dog for sodium. Now I am going to see some terms. such as osmoles and osmolarity. Osmoles and osmolarity. So osmolarity is milliosmoles per liter of solvent. Milliosmoles per liter of solvent. Normal plasma osmolarity is 300 milliosmoles per liter. 300 milliosmoles per liter is the osmolarity of plasma. To remember this fact. So there's a formula to calculate plasma osmolarity. Plasma osmolarity is equal to 2 times the sodium ions plus 0.05 times the glucose concentration plus 0.36 times the blood urea nitrogen concentration. Bit in the formula, but need to remember this. Plasma osmolarity can be calculated by 2 times sodium multiplied by 0.5 times glucose multiplied by 0.36 times blood urea nitrogen. All these values will be given. Simply put the formula. Now I am going to see another term, that is osmolality. Osmolality, which is milliosmoles per kg of solvent. Milliosmoles per kg of solvent. Here osmolarity is milliosmoles per liter of solvent, but here osmolality is milliosmoles per kg of solvent. So it's a very important topics. Don't get terms, don't get confused. Milliosmoles per liter of solvent, milliosmoles per kg of solvent. So this is all about passive transport. Next we are going to see is active transport. Now we are going to see is active transport. Active transport, the first feature is, we know, ATP hydrolysis is required. A for ATP, A for active, seen above. ATP hydrolysis is obviously required in this active transport. This active transport, for this ATP hydrolysis, on the basis of ATP hydrolysis uses, is of two types. First is primary active transport. First is primary active transport. This is primary. And obviously the second is secondary active transport. Secondary active transport. Primary active transport and secondary active transport. Some examples of primary active transporter are universal sodium potassium ATPase. Sodium potassium ATPase. Next is our calcium ATPase. Calcium ATPase. Next one is H plus ATPase, that is our proton pump. Proton pump, as you see in the lysosomes, HPS ATPase or the proton pump. So you see here, all these words contains ATPase, ATPase, ATPase. So these three transporters, ATPase causes ATP hydrolysis. Sodium potassium ATPase, calcium ATPase and H plus ATPase, that is proton transport. ATP hydrolysis is required. Now we are going to see this universal sodium potassium ATPase or simple sodium potassium pump. Sodium potassium ATPase or sodium potassium pump. This sodium potassium ATPase has two subunits. It's a rough diagram, it's not to the scale diagram. Sodium potassium ATPase has two subunits. Subunits being alpha subunit and beta subunit. Alpha subunit and beta subunits are the two subunits of ATPase. Which ATPase? Sodium potassium ATPase, alpha and beta subunits. So this is why this is called a dimer. Because these two subunits is called a dimer. These are two different types of subunits, so also a heterodimer. It's two different subunits, so a heterodimer. Alpha subunits has further such forms, subforms such as alpha 1, alpha 2 and alpha 3. Similarly, this beta contains also some subforms, beta 1, beta 2 and beta 3. So to remember this, it's called a dimer or a heterodimer, this sodium potassium ATPase. Now we are going to see how sodium potassium ATPase functions. This is the alpha subunit, this is the beta subunit, beta subunit. Sodium potassium ATPase, as we know, transports to the outside 3 sodium ions, and for the exchange it transports inside 2 potassium ions. It's a very common fact. We know sodium potassium ATPase transports 3 sodium outside for exchange of 2 potassium inside. So we have a coupling ratio here, coupling ratio of sodium potassium ATPase, which is 3:2. Coupling ratio is 3:2. Remember, 3:2, not 2:3. It's always 3:2, the coupling ratio. As sodium potassium ATPase transports 3 sodium outside and 2 potassium inside, so there occurs a generation of charge or electric charge. There occurs generation of electric charge. So inside becomes negative. Since 3 sodium goes out, 2 potassium is taken inside, so a positive ion is deficient, so the inside becomes negative. As the sodium potassium pump generates a charge, causes a generation of electric charge, which is also called a electrogenic pump. It causes generation genic of electric charge. Electro, it's a pump that puts 3 sodium outside and 2 potassium inside. It's called an electrogenic pump. Important concept we need to understand and remember. Next we are going to see the subunits. This alpha subunit, the 2 subunits, this alpha subunit has binding sites. This alpha subunit has binding sites. These binding sites of alpha subunit are in 2 portions. two portions of this binding site of alpha subunit. To see, first portion is outside the cell or outside the membrane. This is outside the cell membrane. Another one is inside the cell. You see from this diagram, alpha subunit spans across the membrane. This is the outside part, this is the inside part. This outside part contains some binding sites, the inside part contains some binding sites. We are going to see what are those. The outside of the cell is obviously extracellular. This extracellular has potassium binding sites. This extracellular outside the cell is potassium binding sites. Next is this inside the cell or intracellular. This intracellular has sodium binding sites and also ATP binding sites. We have sodium binding sites and also ATP binding sites where inside the cell or intracellular. The extracellular we have potassium binding sites. This potassium binding site, we have this extracellular domain, we have a blocker. This extracellular site has a blocker called Oubain, Oubain. This extracellular binding site has a blocker Oubain. This is similar to digitalis group of agents. Digitalis group of agents. This Oubain is a extracellular part of the alpha subunit blocker. It is the K+ binding sites, similar to the digitalis group of agents. Inside the cell intracellular we have sodium binding sites and ATP binding sites. You can remember this pretty easily. This sodium, 3 potassium, 3 sodium gets transported outside. So this intracellular have sodium binding sites. This potassium, 2 potassium gets transported inside. So this extracellular part has this potassium binding sites. We have discussed the alpha subunits binding sites. Now we are going to see some functions of sodium potassium ATPase. Functions of sodium potassium ATPase. First function is regulation of cell volume. How this regulates the cell volume? We have seen above. If the cell, this is a cell, sodium potassium transports 3 sodium outside, brings 2 potassium inside. So with this sodium, it also expels out some water molecules. This sodium is a osmotically active substance discussed above. So as the sodium gets out, it takes a water with it. This water is known as the H for sodium. So it helps in regulation of cell volume by this process. Next is a very minor contribution to resting membrane potential, which is our RMP. What is this contribution? Very minor, -4 millivolt. Very minor contribution to resting membrane potential, RMP, -4 millivolt. -4 millivolt of all is contributed by sodium potassium ATPase. The third important function is indirect energy source for secondary active transport. You've seen above in the classification of active transport, primary active transport and secondary active transport. Primary active transport, these transport channels which has ATPase activity uses directly ATP. Secondary active transport uses ATP indirectly. This is the difference. Directly uses, so it's primary. It uses indirectly, that is why it is secondary. So this function of ATPase that provides energy for, you can say, indirect energy source for secondary active transport. So now we're going to see this regulation of pump, regulation of this sodium potassium, ATPase sodium potassium pump. The factors which increases activity of sodium potassium ATPase, factors which increases the activity of sodium potassium ATPase are the thyroid hormones, insulin, and our aldosterone. These three hormones, thyroid hormones, insulin, and aldosterone, increases activity of sodium potassium ATPase. Now the factors which decreases activity of sodium potassium ATPase we have Oben and digitalis. We have discussed above that this Oben, which belongs to a digitalis group of agents, is a blocker of the extracellular potassium binding sites. So this Oben and digitalis decreases the activity of sodium potassium ATPase. Next one is dopamine. It's a very important one, dopamine. Dopamine, by inhibiting or decreasing the activity of sodium ATPase, causes natriuresis in kidneys. This dopamine, by decreasing the activity of sodium potassium ATPase, causes natriuresis in the kidneys. This thyroid hormones, insulin and aldosterone, increases the activity. Oben and digitalis and dopamine decreases the activity of sodium potassium pump. So this is all about our discussion on primary active transport. Primary active transport directly uses ATP. Now we are going to discuss secondary active transport. Secondary active transport. First, the feature of the secondary active transport, we need to remember that there are two or more substances are transported. Two or more substances are transported in the secondary active transport. These two or more substances can be of two types. They can be transported in same direction. Two or more substances can be transported in the same direction or in obviously opposite direction. The same direction, two or more substances can be transported, or in the opposite direction. Same direction are called co-transporters. Co-transporters or symporters. Sim in the same direction, co-transporters or symporters. In this opposite direction, these are called the exchangers. Two or more substances are exchanged, that is in the opposite direction. Exchangers or antipodes. imports two or more substances in the same direction. These exchangers are antipodes in the opposite direction. Examples of these co-transporters are SGLT, stands for sodium glucose co-transporters. Sodium and glucose are transported along with each other at the same time in the same direction. So SGLT stands for sodium glucose co-transporters. Next one is sodium potassium 2-chloride co-transporter. Sodium potassium 2-chloride co-transporter. Next one is sodium iodide symporter. Sodium iodide symporter, also an example of co-transporter, is important in the thyroid glands. We will discuss further in endocrine physiology. For now, remember the sodium iodide symporter, sodium potassium 2-chloride co-transporter in the kidneys, and SGLT, sodium glucose co-transporters. For the exchangers, we have a common example of chloride bicarbonate ion exchanger. As here two anions are getting exchanged, this is also known as anion exchanger. Called an anion exchanger. Chloride bicarbonate exchanger. Example of exchangers or the antipodes. So we are going to discuss this example of sodium glucose co-transporter, that is SGLT, sodium glucose transporter co-transporter. How this is going to function, we are going to see. Suppose this is a cell, this is a cell, we have the cell outside here is a vessel or a tissue. Supposedly this is a blood vessel, and this is a blood vessel. The end of the cell, which is near to the blood vessels, is called basolateral end. This is basolateral end. End close to the blood vessel is known, the cell is known as the basolateral end. This end, which is away from the blood vessel, is called of the apical end. So this is the basolateral end, and the away one is apical end. So through this apical end, we are going to see what happens. This apical end, sodium ion moves inside. Listen carefully. Sodium ion moves inside. Along this sodium, this glucose ion follows. This glucose ion follows the sodium as they both move inside. In this intracellular sodium is low. So the sodium which is high outside near this apical end, and the intracellular sodium is low, that's why it moves inside. Along with it, the glucose follows. So as they follow, here we have this transporter. What is this transporter? Our sodium potassium ATPase. You know, the sodium potassium ATPase transports 3 sodium to outside. So this transports 3 sodium from the cell to the blood vessel in exchange of simple 2 potassium inside. 2 potassium inside. So for the sodium that comes inside the cell, this gets transported outside to the blood vessel in exchange of the 2 potassium that comes inside the cell with the help of sodium potassium ATPase. Now we know sodium potassium ATPase causes ATP hydrolysis. ATP hydrolysis. Sodium potassium ATPase, we know this causes ATP hydrolysis. So we came to the conclusion that secondary active transport utilizes gradient established by primary active transport. We have seen here the hydrolysis of this ATP to the sodium potassium ATP is an example of primary active transport. This creates the gradient for the movement of sodium inside, through which the glucose also follows the cell inside. So we are going to conclude this with a flow chart. Three sodium ions transported out through sodium potassium ATPase. This lowers the level of intracellular sodium becomes low. This lowers the level of intracellular sodiums. This creates a gradient. A gradient is created. Outside sodium, which is in higher concentration, moves inside the cell. The gradient is created. This outside sodium, which is in higher concentration, moves inside the cell. Glucose follows through it. Glucose follows this sodium. This is the conclusion of the whole drama. Cytolysis of the sodium potassium ATPase, that is 1 degree transport, create this gradient which leads to this sodium glucose co-transport, which is a secondary transport. So this explains the fact that secondary active transport utilizes the gradient established by primary active transport. Here most important point is ATP not directly used. Is directly what is important, ATP not directly used, where secondary transport. So we are going to see some sodium glucose co-transporters or SGLTs. First is SGLT1. They are present in intestine. So that's why we use ORS for treatment of diarrhea. It's an important question often asked in exams. We use ORS for treatment of diarrhea. Why? Because in ORS we have sodium and glucose. These two are co-transported with the means of SGLT1 or the sodium glucose co-transporters in the intestine. In diarrhea, we tend to lose sodium potassium as well as glucose. For this, ORS contains sodium and glucose which are co-transported in intestine with the help of SGLT1. It's simple understanding. Now, second one is SGLT2. This is SGLT1, this is SGLT2. SGLT2 is in the PCT of kidneys. These two locations we need to remember: SGLT1 in intestine and SGLT2 in the PCT of kidneys. SGLT2 we further discuss in the renal physiology. For time remember, SGLT2 is in PCT of kidneys. Now we are going to discuss a special type of transport here, which is called vesicular transport. Vesicular transport. Vesicular transport, obviously from the name, vesicles are required. Vesicles are required. We know from the cell organelle discussion which vesicles are found in where? Golgi apparatus. Golgi apparatus vesicles are found. This vesicular transport, vesicles are required. This is a form of active transport. Very, very important. Vesicular transport is a form of active transport. Active transport uses ATP. Obvious fact. Vesicular transport is a form of active transport, uses ATP. We are going to see some types. So this is a cell. This is a vesicle actually. This is a vesicle. It's transported outside. This is another vesicle. It's transported inside. This outside transport is called exocytosis. Transport of the vesicles outside is exocytosis. This transport of inside vesicles is endocytosis. Simple concepts, nothing fancy here. Exocytosis and endocytosis. Outside the cell, inside the cell. What are transported? Vesicles. This transport, important, remember, they require calcium. Vesicular transport, the vesicles for the transport requires calcium. To see some special proteins required here. Special proteins required for exocytosis. For exocytosis, we have the snare proteins. For exocytosis, we have snare proteins. For endocytosis, we have this protein called clathrin. Clathrin protein. Also megalin. For exocytosis, we have snare proteins. For endocytosis, we have clathrin and megalin. These two special proteins we need to remember. You need to remember that all the vesicular transport requires calcium. Last important point is no role for concentration gradient. There is no role for concentration gradient as it's a form of active transport. It uses ATP, so there is no role required for concentration gradient. We are going to see some forms of endocytosis. Forms of endocytosis. Important discussion, listen carefully. Suppose this is a cell. This is cell. I'm going to see three types of endocytosis here. First one, see this is here is a, for example, a microorganism. Here is a microorganism. This microorganism gets this word, this engulfed. This microorganism gets engulfed for destruction. Cell engulfs this microorganism outside for destruction. This is called, known as phagocytosis. It's simple, phagocytosis. This microorganism gets engulfed for destruction. Next is, suppose this is a fluid filled vesicle. This fluid filled vesicle gets pinched off inside. This fluid filled vesicle gets pinched off inside. This form of endocytosis is called pinocytosis. Pinocytosis, in which a fluid filled vesicle gets pinched off inside. Phagocytosis, the microorganism is engulfed for destruction. Third type of endocytosis we are going to see. This is, suppose this is a receptor. This is a receptor. Suppose this is molecule of LDL cholesterol. So receptor, molecule of LDL cholesterol. To this receptor for LDL cholesterol, two molecules are attached. What are these two molecules? Molecule 1 and molecule 2. These two molecules are attached. This outside molecule is dynamine. This outside molecule is known as dynamine. So this arrangement is in the form of a triskelion. The other protein involved here is clathrin. I've seen this clathrin protein here. For endocytosis, this clathrin protein is utilized here in this triskelion arrangement, where this dynamine protein is also present. Through this LDL cholesterol gets transported inside. This is called receptor mediated endocytosis. This is called receptor mediated endocytosis. We've seen three types of endocytosis: phagocytosis, pinocytosis, and receptor mediated endocytosis. Receptor mediated endocytosis is very important in lipid metabolisms. I want to discuss much more in details in the lipid, cholesterol, LDL, HDL metabolism in biochemistry. So no need to worry. Just remember, this is receptor mediated endocytosis in lipid metabolism. Another form of endocytosis I want to see. Suppose this is a cell. This is a molecule. It gets transported inside a vesicle, to be accurate. Simply called endocytosis. Molecule gets endocytosed, then again it's transport outside, called exocytosis. Molecule gets transported again outside. This transport of vesicle across the cell, transport of vesicles, called transcytosis. This another example of this vesicle transport across the cell membrane called transcytosis. We have seen these three types of endocytosis: pinocytosis, phagocytosis, and receptor-mediated endocytosis. Now we are going to see some types of exocytosis. Exocytosis are also called emiocytosis. Exocytosis we have seen above requires snare proteins. It requires the snare proteins. The exocytosis require clathrin and megalin protein. Exocytosis, snare proteins, special proteins required. These snare proteins contains a protein called synaptobrevin. Very important protein. Snare protein contains synaptobrevin. The synaptobrevin is required for exocytosis of acetylcholine. From this word you can imagine the synaptobrevin protein is present in the synapse. Synapse is in between the two neurons. Synaptobrevin is present in this synapse. So synaptobrevin is a type of snare protein, is required for exocytosis of acetylcholine. Acetylcholine is the neurotransmitter required for the transmission of nerve impulses from one nerve fiber to another. It is required for muscle contraction. This exocytosis of acetylcholine is very important for muscle contraction. Now, what's the problem? I'm going to see. Now, it's a toxin called botulinum toxin. This botulinum toxin, it degrades the synaptobrevin. This botulinum toxin degrades the synaptobrevin, so it inhibits acetylcholine release. Synaptobrevin is required for exocytosis of acetylcholine, so degradation of synaptobrevin inhibits acetylcholine release. This leads to flaccid paralysis. This inhibition of acetylcholine release leads to flaccid paralysis. This toxin, botulinum toxin, degrades the synaptobrevin, leading to flaccid paralysis. Synaptobrevin is required for exocytosis of acetylcholine. It's a type of snare protein. So this are important some clinical discussions. For the last part, we are going to see some graphs. It's transport mechanisms requires important discussion on graphs. So I'm going to have y-axis and x-axis. So y-axis, this is x-axis, pretty common. Now we have on the y-axis we have rate of transport. active transport represented on the y-axis, and x-axis represents concentration of substance. X-axis represents concentration of substance. So we have two graphs. This is the first graph, which is a pretty straight line. Sorry, can't draw a straight line, but this is a straight line. Straight line denotes simple diffusion. Simple diffusion. Straight line continues, not simple diffusion, and it show no saturation kinetics. As there are no proteins involved in this simple diffusion, and proteins tend to get saturated, the simple diffusion shows no saturation kinetics. This is a straight line graph for simple diffusion. The next graph we are going to see. This next graph continues straight upward line for some moment. After this follows a plateau. Plateau phase. This plateau phase is due to, remember, saturation of proteins. Due to saturation of proteins. This graph shows saturation kinetics, which requires carrier proteins and channels. Any form of protein which involved shows this graph. This is a plateau phase which is due to saturation of proteins. Example, active transport uses different forms of ATPase. Then vesicular transport, vesicles are used, which are proteins. Then we have this passive transport. Can you guess? Yes, facilitated transport. Facilitated transport uses proteins. So as it uses proteins, it follows is saturation kinetics. There is an initial phase of growth, initial phase of increase in the rate of transport, but after, due to saturation of proteins, follows a plateau phase. So these are the examples of this graph. So with this, we conclude this discussion of this cell transport. We have seen passive transports, types of passive transports, and diffusion and facilitated diffusion. Then seen active transports. There are primary active transport and secondary active transports. Examples of primary active transports, the ATPases. Secondary active transports, which indirectly uses ATP, the gradient created by ATP, such as SGLT, sodium glucose cotransporters. Then there is vesicular transport, which is important. It's a form of active transport and it uses ATP. And then the graphs, which follows what kinetics is a straight line or after some time follows a plateau, the examples. Next, to revise this topic, very easy topic, nothing much detail to be understood. Thanks for watching. I will catch you in the next one. Good luck.